Is this working? No. Now it does. Okay, so before I left you that we have decided that we want to record the activity of a specific population of neuron. We have used viruses to deliver our genetically encoded calcium indicator in the brain so that these neurons get infected. They start to express the genetically encoded calcium indicator, and we are able to see their activity by the changes in calcium concentration. Another thing that we have done is to perform a surgery on the skull to remove a small portion of the skull and change it into a piece of glass so that our skull is literally transparent now, or basically there is no skull, but there is a material that is transparent that will allow us to basically see fluorescence from within the brain. So now we need to visualize calcium dynamics, and of course we need a microscope, right? No brainer. But which microscope do you use? So there are three different needs that we need to satisfy in this case. We need to be extremely precise in space. I told you before that the brain is a very dense tissue. There are neurons all over the place. All of these neurons will have different calcium dynamics and different things, and we should be able to precisely associate and assign change in fluorescence in the pixels that we record from a specific neuron. So we need to have very specific in space. We need to record a lot of photons because these neurons are very small, and the signal that we get is very, very dim. So we need to record a lot of photons. And we are trying to do that, so to have light go in and out of the brain in a tissue that is very, very fat. And that fat means scattering of photons. So basically reflecting photons all over the place in every direction. Which means that we are going to be able to reach just very few, like micron below the position of our surface, unless we use some trick. And this is the trick that we use. So the first thing that I want to tell you is I want to remind you how fluorescent works. I mean, you should have seen these concepts already during your sum of blockers of your studies. Please not with your head because I cannot go through optics again. Okay, perfect. But one thing that I just want to tell you is, do you know already about two-photon excitation? Maybe I'm just under thing. Okay, so I'll go fast in that. So usually you have to provide certain energy to the molecule that is excitable, so that the electrons are jumping to the excited states. They release a little bit of energy to go to the baseline, basically level of the excited states, and then when they release energy to go back to the ground state, they emit some fluorescence. So you give a photon to excite the neurons that is at a smaller wavelength because you need to give them more energy. And then you get a photon that is emitted back by this molecule at a longer wavelength because it has lost a little bit of energy in this transition, right? Now, the principle of two-photon excitation is that to create this jump from the ground state to the excited state, you don't use just one photon. You don't give the energy for kicking them up to the excited state with one photon, but you do it with two photons. So basically you provide half of the energy, which means double the wavelength, or more or less, so that the neurons basically, only if they are able to absorb two photons in a very small window of time, we're talking about femtoseconds, they are able to be excited and to go to the excited state and then release and emit a certain amount of fluorescence. Why is this important? The important is that the problem in this case is that the visual state is extremely unstable. So this state here is extremely unstable, and the two photon needs to be absorbed almost instantaneously. We're talking really about very, very small delays, femtosecond delays. The answer that you give to that is to develop lasers that are able to pulse packets of photons with very high frequency, so that the probability that these two photons are absorbed at the proper delay of time is actually high, and you can get excitation to the excited state, right? The advantage of using this is that by using two photons instead of one, you solve basically all the three problems that I was telling you about in one go. In one case, because the long wavelength suffered less from scattering. It penetrates through the fat tissue for more amount of space. So it means that you can actually image deeper within the brain that you would normally do just with one individual photon. The other thing is that because this two photon needs to be absorbed at the proper temporal delay and a very short temporal delay, the optimal temporal delay for the two photon effect is going to be at a very specific focal plane, a very specific point in space. If you compare a one photon microscope versus a two photon microscope, you see that while the one photon microscope also molecules that are above and below the area where you actually want to image are excited by the photon. That's why you actually use a pinhole in confocal microscopy. In this case, only the molecules that are at the focal plane are able to be excited and then to emit fluorescence. So you gain a lot of precision by this, and you are laser sharp. Sorry for the joke. The third thing is that you can recall and get more light just with excitation with two photons. You can focus more because you don't use a pinhole. So you basically do not cut down photons, but whatever you get that is emitted from this tissue is emitted from the specific focal plane where you're actually imaged. Of course, there are some caveats. This is not an instantaneous plane. However, two photon microscopes are really big and large pieces of equipment. This is a picture from the one that we have in our lab. This is the one that you would come and see if you were able actually to come and do your block course, as you should have done the block course. But there is a lot of components that go in this. This is basically taller than me. And I mean, I'm quite short, but still it's taller than 2 meters when you put it on the table and so on. So they're incredibly big type of equipment. So what happens usually is that because the objective is basically fixed and you don't want to have motion, the animal cannot just go in and out of focus. What you do is that you basically clamp the mouse head here through a head fixation system. The mouse is able to do something under the two photon, which depends on the type of dynamics or the type of phenomena that you want to investigate. And then at the same time, you record activity from the neurons that express the calcium indicator and that you have made visible by your craniotomy, or let's say your cranial window, under the microscope. And the type of behavior that the animal does under the microscope largely depends really from what you want to see. If you want to analyze intrinsic responses of neurons, or to which sensory stimuli they respond to, or the intrinsic activity of the neurons, the tuning curve, the receptive fields concept that we're going to go through tomorrow, you can have a net fixed mouse that is anesthetized under the microscope while you deliver some sensory stimulation. This is partly the first type of application for two photon microscopy that has been done now almost 15 years ago. If you want to record the response of neuron in alert animals that are learning, making decision, or predicting what is going to happen in their future, you need a mouse that is head fixed under the microscope, but it is also awake and behaving. And we are going to see in the next slides how you can actually achieve that. In that case, you need to add the complication that you need to synchronize the trace of your cell activity to the trace of the mouse behavior. And this is something that we're going to go through tomorrow. So I'm just going to leave it for tomorrow. The third thing is that you might want to investigate a phenomenon that is really impossible to be done in head-fixed animals. So the animal has to move freely. For that, it is possible to actually investigate this phenomena. It has become possible only recently. We're talking about the last 7-8 years. There has been a tremendous technological effort to miniaturize microscopes, both one photon and two photon microscope, to make them so tiny that you can actually implant them or fix them on the head of the animal, and the animal carries them around when they're moving in a box. This is another thing that you should have seen by coming into the lab. You cannot see, but when the virus is over, if you're curious, just come and we'll show you some of those experiments. But let's go back to point number 2. Imagine that the type of phenomenon that you want to create does not suffer by the fact that you are impeding some movement of the head of the animal. In that case, you can synchronize the recording through your microscope to a virtual reality setup that is able to let the animal move freely when its head is actually fixed and not moving at all. So what happens is that the animal usually is head fixed under the microscope, but is basically located either onto a floating ball or to a linear treadmill that can actually move, so that the animal can run on the treadmill or on the ball without physically moving because it's head fixed. And the signal that is picked up by the movement of the ball or the treadmill is used to create a closed loop with a projector that is able to project the image of the visual flow on a circular dome in front of the animal. It's basically a cinema for the animal that runs, that moves, is able to manipulate the visual flows that it sees so that it has the impression of running into a linear track or into an open field. And one example of this is in this movie here. I just want you to see. You see here the animal running on one of these balls, the visual flow moving in sync with the movement of the ball, which of course is manipulated by the animal, and the animal going under these cues here, because these cues is where the animal gets some sort of water or sugary reward. So it likes to go there because it gets some reward, and you see that the animal could be moving in this arena here, but it really likes to go where there is a cue. So these are different type of virtual reality setup that have been implemented now for the past 10 years for studying phenomena like navigation, like memory, like the formation of specific representation, like the ability to accumulate evidence and make decision based on the evidence that you have accumulated. They're quite powerful. We have some of these setups in the lab. Stefan, Kevin, and Daniel are building some other of these setups. They're really demanding from the point of view of the engineering, let's say, but they can be done and they can be used to do something quite exciting. If your behavior requires the animal to move freely, there is no alternative at the moment. Then, if you want to do calcium imaging, of course, then doing something like this. So to miniaturize a miniscope, to use a small LED source that is able to be delivered in the brain of the animal through a small lens, which is in close contact with the preparation of the animal, and to fix this small setup that is basically like 2 grams small on top of the head of the animal. You see here one of our of the animal that we actually, this is one that I recorded when I was doing my postdoc, but we do have the same here in the lab, in which the small miniscope is actually fixed on the head of the animal. The animal is moving in a big box of 1 meter by 1 meter. And here you see the movement in real space. The animal is moving. You see here the LED that excites the neurons and that makes you able to record activity of individual neurons while the animal is freely moving. This version of the miniscope does not use 2 photons. It uses just 1 photon because the 2-photon laser is a beast. Right now, the newest development is that there are also miniscopes that work with the 2-photon effect, so that you can combine the advantage of having 2 photons with the advantage of having freely moving. One advantage of having 2 photons is what I told you before, but also the possibility that with 2 photons, you're able to image finer details of the neuron. So a lab in Munich can actually done a comparison between the type of resolution that you can get with the 2-photon microscope that is on your right with the one that you get with the 2-photon miniscope. And you see that the resolution is really different. You lose a lot of resolution in 1 photon, both in X, Y, and Z. And here you can see how you see single cells with very nice dendrites. You can arrive and increase your resolution to get even spine details. But this is something that is basically impossible to do with the miniaturized 1 photon. So you use the 2-photon microscope, you use the 1-photon microscope to pick up the calcium traces to get movies of neurons that are active or during specific behavior, like the movie that I showed before. And at this point, and this is where our presentation of today sort of ends, you end up with movies of cells blinking that you need to make sense of. And this is where a complex part of image and data analysis starts. You need to process the movie so that you can remove any small motion artifact, because your cells go in and out of the plane of focus. If you've ever been at the microscope, you know what I'm talking about. The heart is beating, the mouse is breathing, it's doing small movements, and they all impact the way in which the cells actually are moving. Your brain moves into your skulls to an amount that you would never consider possible. This is one of the biggest problems that you do every time you start to do some surgeries on the brain. So the first thing that you do is you register the motion, so align all the frame of your moving one to the other. You use some algorithm to identify the footprint of the neuron. What I mean is that, for example, in this case, you see that this is a very active neuron. You create algorithm to select and create basically a contour along this neuron so that you can quantify the mean change in fluorescence in the pixels that are within this neuron here, and calculate how much of this change in fluorescence come from the cell, from how much sensing fluorescence come from different sources of noise. That can be the other cells around, the axons that are close by, the dendrites that are close by, and so on and so forth, which is called neuropil. At that point, you use the calcium traces that I showed you before. One example you see it here, these different calcium traces, to try to infer when the neuron was active through the process of deconvolution. Remember, this is not a direct measure of activity. And in the end, you check if your segmentation algorithm has done a good job. By that moment, this is an example of one of those movies. You have a movie in which the different cells are blinking. You use your network to identify individual neurons. For example, these red highlighted areas here, 1, 2, 3, 4, 5. You're able to calculate the calcium trace of each individual neuron by just quantify the change in fluorescence over time. You align this change in fluorescence over time to the specific behavior that the animal is doing. In this case, it's just running along a linear corridor in the virtual reality. And then you can quantify if specific cells are able to express specific pattern of activity that we consider and we call turning curves. And this is the topic of tomorrow. So we are going to pick it there to try to make sense of what neurons are actually saying when they're active, when the animal is doing anything. Most of the time, what you end up is something like this, is the same thing that we ended up yesterday, if you remember. So you end up with a raster plot of the activity of individual neurons over time, and you have to try to make sense of what each of these neurons is trying to say when the animal is doing something. And how do you do that is the topic of tomorrow's presentation. So to wrap up what we went through today, and this is the beginning of our end of the day discussion, we know that neurons connect to each other, and the way in which they connect to each other influence who they speak to, what they have to say when they're actually talking. We are talking about this as the temporal dynamics of activity of individual neurons and individual neuronal networks. The topology of the neuron, meaning the way in which the individual circuit motifs are combined to each other, defines the temporal dynamic of a neuron activity, and you have seen this in your examples. This temporal dynamic defines which messages are strengthened and which one are weakened, because they define and basically select which neuron are coactive, and on which synapses you can apply the rules of LTP and LTD. And calcium imaging can be used as a method to record the activity of large population of neurons from genetically identified neurons and over a long period of time. Because of course, the animal can be put under the microscope over and over and over again over the course of different days, until of course the animal is still compliant to you and it wants to behave under the microscope, and until the cells, as we have seen from the question that we have received through Slido, don't become sick, and basically you cannot infer any good information from that anymore. So this completes the thing that I wanted to say about the way in which you do calcium imaging and the way in which you can record the activity of neurons. The first part, these first 2 days, were quite technical and technically oriented. I just gave you sort of an introduction of how we can actually try to understand what the brain is doing, what single neurons are saying, what single neurons encode for. That is the topic of tomorrow, of Thursday, and partially also of our Fridays lecture. So we're back at the one-to-one. Do you have any question about today's presentation? Let me see if there is anything anonymously. No, we are stopping there. Any question about the exercises or comments about the exercises of today? No. Who was Vilde was bringing something up, right? That you encountered a strange artifact during your in your group during your exercises.
Yeah, we had one observation about the refractory period, so I can share the screen. Which basically is that, can I? Yeah, there I can move it. Good.
Yeah.
So what got pointed out is that if you put the refractory period to be 0 and you have a increased current output, then you get this kind of alternating pattern, or not alternating, but waveform pattern.
Yeah, so I don't think that in this case, this is really biological. You're pushing the situation to a very naturalistic situation. So the answer of this exercise is that if you don't have a refractory period, you're basically not putting a upper limit to the firing frequency that the neuron can achieve, right? Because in that case, it doesn't, usually neuron needs to wait for the refractory period to produce another spike. And if you have a continuous accelerator injection current like the one that you're doing now, but you don't have a refractory period, the only limit to the creation of a second action potential is that the first action potential is terminated. So I think that this is what you're seeing right now. So the spikes are coming at the frequency that is basically just to the end of what they normally would do, just coming back to the level of the threshold and then shoot up again because you're injecting current. I don't think that this is something that you would ever be able to experience in a naturalistic condition. It might be just a quirk of the model that you've pushed too far of the explanation. I don't think that this would be real. So I don't know how if this is like a real explanation to be given below.
I mean, today picture that was just shared is also a weird thing about the one that with the refractory period, because if you look at the part where it should go into the hyperpolarization, there's like two peaks.
Mm-hmm.
I don't know if you can see it.
In this image?
And in the one that was just currently shown.
Yeah.
I can share it again.
Yeah, that other peak it's weird. I would say that is a strange simulation. I haven't seen it before. You've also changed the voltmeter parameters, right, for the visualization. I see that that's the one that you have opened there.
It was just default.
Okay, is the default isn't changed? No, then yeah, I would say that it's just this is an application, and it might be just that you're pushing the simulation to the test of where it gives you something that is biologically meaningful. Anyone else saw something interesting? Or maybe combined all the different connectivity motifs in one super network that is going to be smarter than anyone in the room? Anybody wants to share his or her creativity of today? So tomorrow we're going to go through more complicated networks, and you have got some example of these in the files that you get on Adam. We're going to talk about particularly some of those and the influence that they have on the way in which our brain represents space, for example. So this is going to be something really close to what we do in the lab and something that I think is really cool. So yeah, I'm looking forward to that. Okay, last instance for asking questions for today. Otherwise, I'll send you on your way and bid you farewell for tomorrow. Great. So a lot of delta waves approaching. Guys, see you tomorrow, and we'll start again at 9:30, right? Like today. So any problem, comments, or stuff, you can still write me a message or even write on Slido. I think that this is actually going on overnight. So